أعوذ بالله من الشيطان الرجيم بسم الله الرحمن الرحيم ولقد فجرنا الأرض عيونا فالتقى الماء على أمر قد قدر وحملناه على ذات ألواح ودسر تجري بأعيننا جزاء لمن كان كفر صدق الله العلي العظيم. Lecture number 21, 9th class. Classification and classification of the living thing is my topic these days, because this chapter is telling you how the living things are being classified from kingdom to species. You are starting from phylum, you are going to species, and these slides which I am showing you today, you will keep these slides with you, because these are the key points of this chapter, and these points will help you in learning this chapter very easily. I can send you details. I will tell you my contacts after this clips, and you can contact me, and you can if you want these slides to me to send you, I can send you. Euglena and the bacteria was difficult to adjust in the groups. So these were the main discussions, these were the problems in this chapter, because when you was classifying bacteria was the prokaryote, and when you was classifying the Euglena says I am not, Euglena says I am not plant. When we talked about Euglena, if you are not plant, you are animal, he said no, I am not animal. Mean Euglena having chloroplast will not produce the food. When any organism will not produce the food, that is heterotroph. When any organism will produce the food, that is autotroph. So Euglena is heterotroph. Question is that Euglena is heterotroph? No, that is not heterotroph. So Euglena is autotroph? No, that question is not valid. So that question is also not valid. So Euglena is going to between living plants and animals, mean even having chloroplast, it is heterotroph. So showing tremendous behavior of having chloroplast but without producing food, will not produce the food. So Euglena, I am talking about that Euglena is the between animals and the plants. We go to another thing which I was discussing about the bacteria. Yes, the bacteria is a prokaryote. It was very difficult because prokaryote mean without well-defined nucleus, About the bacteria, about the bacteria and about the Euglena, adjustment was being solved by the scientists. And how they solved, they adjusted Euglena in a kingdom which was having multiple diverse living things. Means you can adjust any student in the college who is not suitable to admit in 9th class, who is not suitable to adjust in 8th class, who is not able to suitable to adjust in all classes of any college. So what you will do, you will make any extra class, and you will say I was telling X class, so I will use that word once again, X class. So Euglena was being adjusted in that phyla in which such organisms are not able to adjust in any other phyla. Euglena. What are the unique things about the Euglena? It is having the chloroplast, and it cannot produce the food. So that was the thing. Euglena become precious, important. Why it became important? Even having chloroplast, it is unable to produce the food. When unable to produce the food, so this is heterotroph. So this is tricky. Even having chloroplast, it is heterotroph. So adjustment in the protista. Go back to bacteria. Difficulty in the bacteria, because bacteria don't resemble even any group. Bacteria don't resemble fungi. Bacteria don't resemble kingdom Animalia. It don't resemble kingdom Animalia Plantae. It don't resemble even the Euglena, because it has without well defined nuclear membrane. Better, if you are taxonomist, better leave bacteria classification, because bacteria did not adjust in the 5 kingdom system of classification. If you want to leave, you can leave, but you will not leave bacteria, because bacteria are the major parasites. Bacteria are the major parasites. They are the major parasites who cause the disease, and these are the parasites who will do cause different type of diseases. And for human beings, if we want to adjust the human beings as a healthy humans, and we want bacteria should not cause the disease to the humans, so what we will do, we will study bacteria DNA should be studied, bacteria plasmid should be studied, and bacteria classification should be studied. Therefore bacteria classification is also very very important for taxonomists and systemics, and they arranged a separate group. What was the name of the scientist? E. Chatton? Yes, E. Chatton. Okay, what was the name of the group? They have given to the bacteria prokaryotic in that time. Currently the name is the prokaryote. Okay, so it is very difficult to adjust in the group bacteria group was different group was made prokaryotic. Yeah, prokaryote group is also called as the Monera. It is also called as a Monera. So now we go back to another slide. Bacteria is a prokaryote. Yes, this is so crucial. This cell is so crucial that the prokaryote without well-defined nuclear membrane, and there are so less types in the world, but with minimum and maximum numbers, that their study is so difficult due to their microscopic nature. But I was telling you there was bacteria named Escherichia coli. Escherichia coli was the bacteria. Escherichia coli was the bacteria who caused a disease in human. Scientists studied Escherichia coli and bacteria. By studying bacteria, they identified that bacteria has a structure called plasmid. Why study of the bacteria is more important? Because bacteria is a prokaryote, it is a parasite, it causes a disease, and studying of the bacteria will help us in understanding the bacteria in the biochemical machinery. We are able to study plasmid because plasmid is a very useful DNA in bacteria. Bacteria consist of two DNA. One type of DNA which is present at the center of the bacteria, one type of the DNA which is present in the cytoplasm of the bacteria. The useless DNA of the bacteria is called plasmid, and that plasmid which is present in the bacteria is used in genetic engineering. You may imagine if you are unable to classify the bacteria into classification in a prokaryotes or Monera, if you are unable to classify the bacteria, you are unable to produce the insulin. We are unable to produce the insulin because insulin is produced by using plasmid of the bacteria by the genetic engineering. Go back to another point, which fungi is between animals and the plants. This is amazing. Euglena is also between animals and the plants. We have to distinguish these two terms, fungi and the Euglena, because you may say these two should be in one group, because fungi is also not animal, not plant, having some characters of animal, having some characters of the plants. Even the Euglena having some characters of animal, some characters of the plant. They should set in same group. But problem, fungi is more modified organisms. Fungi says I am more unique than the Euglena. Fungi say my mode of nutrition is heterotrophic mode of nutrition. Fungi say my mode of nutrition is absorptive mode of nutrition. Absorptive mode of nutrition. What is absorptive mode of nutrition? Absorptive mode of nutrition is that absorptive mode of nutrition is that digestion take place outside the body. Two type of digestion I will explain, not necessary here, but just to want to give you a clear concept of heterotrophic mode of nutrition, digestion which takes place inside the body, that is called intracellular digestion. Intercellular digestion. The digestion which takes place outside the cell, outside the body or outside the cell, that is called extracellular digestion. So what happens in extracellular digestion? The digestion which takes place outside the body. How the digestion take place outside the body? Fungi do extracellular digestion. How it do? It secrete enzymes outside, the food is digested outside, and that's why this absorptive mode of nutrition absorptive mode of nutrition, differentiate between Euglena and fungi. So competition between fungi and Euglena. Euglena says I am more developed, I am more unique, not developed, unique. Fungi say I am more unique. Euglena say I am more unique because I have chloroplast but I cannot prepare the food, so I don't come under category of plants and animal. Fungi say yes, of course I have the cell wall, but my cell wall is composed of the chitin. And now Euglena says I can produce the food sometime in the night, but normally I am heterotroph. Fungi say my cell wall composed of chitin. Normally plants cell wall composed of the cellulose. Fungi says my way of life is absorptive mode of nutrition, which is totally different from in any other organism. Euglena is unable to answer. Fungi says if you can do absorptive mode of nutrition. The Euglena, fungi says to Euglena, I have absorptive mode of nutrition, or in your book written heterotrophic mode of nutrition, a way by which food is digested outside and then enters into body. Fungi says if you are able to eat like that, to feed like that, even any organism, of course fungi is right, because no one living thing eat that food by this way, mean absorptive mode of nutrition. So these two groups were made, and they actually made the difference between kingdom Animalia and kingdom Plantae. Earlier scientists were thinking there were two only main kingdoms, one is the kingdom Animalia, one is the kingdom Plantae. But after the introduction of the fungi and the prokaryotes and Euglena, it looked like that the world life is more than animals and the plants. So animals and plants, of course they are integral parts, never mean that they are the only. So modern classification is based upon the biotechnology, upon the genetics, upon the most diverse kingdom is the Protista. Most diverse kingdom is the Protista. It is based upon the biotechnology. It is based upon the biotechnology and the genetics, and the most diverse kingdom is the Protista. Because two things I told you, the 5 kingdom system of classification can be modified on the basis of these two terms, biotechnology, and what are the name of the two scientists who modified and finally introduced 5 kingdom system of classification, which we are studying currently. That is the, yes, you can speak the name of two scientists whose 5 kingdom system of classification are successful and we are studying now. Margulis and Schwartz. You are right. And which diverse kingdom is there? Yes, the Protista. This is the Protista, and this kingdom is a diverse kingdom. Protista is a diverse. These slides you will keep with you, my sons, because whenever you are classifying the 5 kingdom system of classification, because normally the people think Robert Whittaker 5 kingdom system of classification is a useful, but I will tell you Robert Whittaker 5 kingdom system of classification was being modified by Margulis and the Schwartz, and they based upon these two terms, the biotechnology, they based upon these two terms, which is a pea plant. Pea plant scientific name was the Pisum stivum, and also they based upon the DNA knowledge, they based upon the genes knowledge, they based classification upon the knowledge discovered by the law of independent assortment, law of segregation of the Mendel, and they use the pea plant. Mendel used the pea plant, and that knowledge was being applied in the classification. Most diverse kingdom system is a Protista, mean that any organism which is not going to adjust in any group, you will adjust in the Protista. So Margulis and the Schwartz, yes, of course, they are the heroes, and they discovered the 5 kingdom. They actually modified the 5 kingdom system of classification, and that slide is very crucial, because if you want to finish the classification, you will finish these two scientists, Margulis and the Schwartz. Full name Lynn Margulis and Caroline Schwartz. So these are the contacts. Inshallah, I hope you will contact me, and if you want to get some little information, and these are my contacts. Okay, this is lecture number 21. Classification chapter is the last chapter about general information about biology, then you are going to enter into right field in a penetrate into concepts of biology, because you will start from cell structure, cell function, cell membrane, cell wall, mitochondria, and then you will go to plants tissues, you will go to hypertonic nature, you will go to digestive system, you will go to circulatory system, you will go to cell division, you will go to enzymes, and you will go to the photosynthesis and respiration in much detail. Even the 9th class, very interesting topics are upcoming. These were general informations still I am giving, because first 3 chapters are these are the informations. You are being trained, that you are able, you are being trained, or you are being trained to study the biology, because you know these are the first type of informations, and after the second information you will enter into the systems, and systems knowledge and cell knowledge, and the topics which I told are very interested, because they are directly linked with your body. Thank you. Allah Hafiz.